This presentation is titled Pharma Water Engineer Interview Questions and Answers. It contains 55 technical Q&A prepared by Pharma Talks. Section 1, Fundamentals and Water Quality. Question 1. What is the role of a pharma water engineer? Answer. The role of a pharma water engineer is multifaceted and crucial for pharmaceutical manufacturing. Primarily, they are responsible for designing, operating, maintaining, qualifying, and continuously improving pharmaceutical water systems. Question 2. What types of pharmaceutical water are commonly used? Answer. Different types of pharmaceutical water commonly used. These include potable water, purified water, water for injection, sterile water for injection, and clean or pure steam condensate. The specific grade of water required depends on several factors, such as the dosage form, the particular step in the manufacturing process, product contact, and the associated patient risk. It's also crucial that the water used meets the site specifications, which must align with pharmacopeal standards and regulatory expectations to ensure safety and quality. Question 3. What is the difference between purified water and WFI? Answer. Here, we're looking at the key differences between purified water and water for injection, or WFI. Purified water is generally used in processes that don't require sterility, such as non-sterile manufacturing, cleaning, and other general uses. In contrast, WFI has much stricter requirements, particularly in terms of microbiological quality and endotoxin levels, because it's often used for sensitive applications like injections. Because of these higher standards, the entire system for generating, storing, and distributing WFI must be validated to ensure it meets the intended use. This validation is crucial to maintain safety and compliance in pharmaceutical processes. Question 4. Why is water considered a critical raw material? Water can come into direct contact with the product, equipment, containers, and cleaning surfaces, which means its quality significantly impacts the entire process. If the water quality is poor, it can introduce chemical contaminants, microbes, or endotoxins that could compromise safety and product integrity. Because of this, it's essential to maintain a tightly controlled system, as water quality can change rapidly and unexpectedly. This control ensures that the final product remains safe and effective. Question 5. What parameters do you assess for incoming potable water? Answer. It's essential to assess several key parameters to ensure its quality and suitability for use. We look at conductivity, which helps us understand a water's ionic content, as well as hardness, alkalinity, and pH levels, all of which affect water chemistry and treatment processes. Turbidity is important too, as it indicates the clarity of the water and the presence of suspended particles. We also measure chlorine or chloramine levels, which are common disinfectants, along with the microbial load to assess any biological contamination. Beyond these, it's critical to consider seasonal variations, since water quality can change throughout the year. We also monitor for specific minerals like iron, manganese, and silica, which can impact both water quality and the performance of our treatment systems. Finally, understanding supply stability helps in planning consistent monitoring and pretreatment strategies. All this information together allows us to tailor the right pretreatment and monitoring approach to maintain safe and high-quality potable water. Question 6. What are the major quality risks in a pharmaceutical water system? Answer. The primary concerns include microbial contamination, which can compromise the water's purity and safety, as well as biofilm formation that can harbor microbes and protect them from cleaning efforts. Endotoxins are another significant risk, as they can cause adverse reactions if they enter pharmaceutical products. We also need to watch out for high conductivity and elevated total organic carbon, or TOC, which indicate impurities in the water. Beyond these, there are other common risks like chemical breakthrough, where contaminants penetrate purification systems, membrane failure, which can reduce system effectiveness, stagnation that promotes microbial growth, and poor sanitization practices. To control these risks effectively, it's essential to have a well-designed sanitary system, rigorous monitoring protocols, consistent maintenance, and thorough trending analysis to catch issues early and maintain water quality over time. Question 7. What is conductivity and why is it important? Answer. Conductivity measures the level of ionic contamination in water, which is crucial for maintaining water quality. When conductivity rises, it can signal several potential problems, such as a failure in the reverse osmosis system, issues with electroionization, resin exhaustion, valve leaks, or even chemical contamination. Monitoring conductivity online allows us to detect any decline in water quality promptly, enabling quick action to address the issue before it worsens. This makes conductivity a vital parameter in ensuring consistent water purity. Question 8. What is total organic carbon, or TOC? Answer. TOC is a crucial measurement that tells us about the level of organic contamination in water. This includes anything from leftover cleaning agents to organic fouling, resin breakdown, or even byproducts from microbial activity. Monitoring TOC trends is especially important because it helps us catch any unusual changes in the system early on, allowing for timely intervention before issues escalate. So, understanding TOC gives us valuable insight into the overall health and cleanliness of the water system. Let's talk about endotoxin. Endotoxin is a component found in the cell walls of gram-negative bacteria. It's important because it can trigger severe pyrogenic reactions, which are basically fever-inducing responses, especially when present in injectable products. This makes controlling endotoxin crucial in water for injection systems, where both microbial contamination and endotoxin presence must be tightly regulated to ensure safety and product quality. Question 10. What exactly is a water quality trend? Answer. This typically includes measurements like conductivity, total organic carbon, or TOC, microbial counts, endotoxin levels, temperature, pressure, and flow. By analyzing these trends, we can spot early signs of deterioration before the water quality actually falls outside of its specifications. This proactive approach helps us maintain safe and consistent water quality by addressing potential issues early on. Section 2. Water generation and pretreatment. Now, let's look at questions 11 through 20 that delve deeper into these topics. Question 11. Describe a typical purified water generation system. Answer. It all starts with potable water, which first goes through a pretreatment process. This usually includes filtration to remove particles, softening to reduce hardness, and chlorine removal to ensure the water is safe and does not damage subsequent equipment. Next, the water undergoes further purification through reverse osmosis, or RO, and sometimes an electroionization, or EDI, step. Question 12. Why do we use a water softener before reverse osmosis, or RO? The main reason is to remove hardness ions, specifically calcium and magnesium, from the water. These ions can cause scaling on the RO membrane, which significantly hampers its performance. By softening the water beforehand, we reduce the chances of scaling, which helps in maintaining the efficiency of the RO system. Additionally, a good water softener not only protects the membrane, but also improves the overall recovery rate, extends a membrane's lifespan, and reduces how often the membrane needs to be cleaned. In essence, the softener plays a critical role in keeping the RO system running smoothly and cost-effectively. When a water softener is exhausted, the hardness leakage begins to increase, and this hard water then reaches a reverse osmosis, or RO, feed. This is problematic because the RO membranes can develop scaling from the hard minerals, which results in lower water flow, a higher pressure drop across the system, and poor rejection of contaminants. To avoid this, it's crucial to regularly review the regeneration process of the softener, conduct hardness testing, and ensure preventive maintenance is consistently performed. This helps maintain efficient system operation and prolongs the lifespan of the membranes. Now, let's talk about why activated carbon is used in pretreatment. Activated carbon plays a crucial role by removing chlorine, chloramine, and certain organic contaminants from the water. This is really important because chlorine and chloramine can cause oxidative damage to reverse osmosis membranes, reducing their lifespan and efficiency. However, it's also important to note that activated carbon can support microbial growth, which means regular sanitization and careful monitoring are essential to keep the system safe and functioning properly. This balance ensures the membranes are protected while maintaining overall water quality. Let's talk about reverse osmosis, or RO for short. Essentially, it's a membrane purification process that uses pressure to push water through a special membrane. This membrane acts like a filter, removing a wide range of contaminants, including dissolved salts, ions, tiny particles, microorganisms, and organic compounds. Because of its effectiveness in producing very pure water, reverse osmosis is a key technology in generating pharmaceutical-grade water, which is crucial for many medical and laboratory applications. Let's talk about some common causes of poor reverse osmosis performance. One major factor is scaling, which happens when minerals build up on the membrane surface, reducing efficiency. Fouling and biofouling are also significant issues. Fouling refers to the accumulation of particles or deposits, while biofouling is caused by microbial growth clogging the membranes. Chlorine damage can degrade membrane materials, and aged membranes naturally lose their effectiveness over time. Other contributors include low feed pressure, which means the water isn't being pushed through the system with enough force. Incorrect recovery rates, damaged seals, or poor pretreatment of the feed water can also lead to system failure. To keep track of performance, we monitor key indicators like flow rates, pressure changes, conductivity, and rejection trends. These measurements help us identify problems early and maintain optimal operation. Question 17 asks, what is double-pass reverse osmosis? Essentially, in double-pass RO, the permeate from the first pass, the purified water that is passed through the membrane, becomes the feed water for a second RO membrane. This second pass further purifies the water, improving the removal of ions and significantly lowering the conductivity. This process is especially important when we need higher purity water or want to ensure stronger protection for downstream polishing steps. So, by running the water through two stages, we achieve a much higher quality of purification than a single pass alone. Let's talk about EDI, which stands for electroionization. Essentially, EDI is a water treatment process that combines ion exchange resins, special membranes, and an electrical current to continuously remove ions from water. What makes it particularly useful is that it acts as a polishing step after reverse osmosis, or RO, to further purify the water. Unlike traditional ion exchange methods, EDI does this without the need for chemical regeneration, making it a more efficient and environmentally friendly option. This continuous removal of ions ensures very high purity water, which is essential in many industrial and laboratory applications. Ultrafiltration is a process primarily used to remove very fine particles, microorganisms, and contaminants with high molecular weight from liquids. One key advantage of ultrafiltration is its ability to act as a final barrier in high purity water systems, ensuring the water is as clean as possible before use. Additionally, depending on how the system is designed, ultrafiltration can also help reduce the risk of endotoxins, which are harmful substances released by certain bacteria. This makes ultrafiltration a critical step in applications where water purity and safety are paramount. Let's talk about why UV treatment is used in pharmaceutical water systems. UV light can help support microbial control by inactivating microorganisms, which is crucial for maintaining water purity. It can also assist in breaking down ozone, and in some cases, reduce total organic carbon or TOC, which improves overall water quality. However, the effectiveness of UV depends largely on the specific wavelength used and the design of the system. It's important to remember that UV treatment is not a replacement for good sanitary design, and the continuous circulation needed to keep the water system safe and compliant. So, UV is a valuable part of the water treatment process, but it works best when combined with proper system design and maintenance. Now we'll be focusing on storage and distribution systems. This section covers questions 21 through 30, where we'll explore the key concepts and mechanisms behind how goods and materials are stored efficiently and distributed effectively. Understanding these systems is crucial because they ensure that products are available when and where they are needed, minimizing delays and reducing costs. We'll look into different types of storage methods, inventory management practices, and the logistics involved in distribution networks. This will give you a comprehensive view of the entire supply chain process, helping you grasp how storage and distribution contribute to overall operational success. Let's talk about why continuous recirculation is so important in a water loop. First, it helps minimize stagnation. When water sits still for too long, it can become a breeding ground for microbes, so keeping it moving supports microbial control and keeps the water safer. Additionally, Continuous recirculation helps maintain consistent water quality across all points where the water is used, so users get clean water every time. Finally, monitoring the return loop gives us valuable information about the overall condition of the distribution system, helping us catch and address any issues early on. Continuous recirculation is really key to ensuring a reliable and safe water system. So, what exactly is a dead leg? A dead leg refers to a section of piping where there is little to no effective circulation of fluid. Because the flow is stagnant or very limited in these areas, it creates conditions that allow biofilms to form more easily. This is a significant concern because biofilms can harbor bacteria and pose hygiene risks. To prevent this, dead legs should be minimized as much as possible. Proper sanitary design and adhering to established standards help ensure that these stagnant areas are reduced, promoting cleaner and safer systems overall. So, what exactly is a biofilm? Essentially, it's a microbial community that forms a protective layer while attaching itself to a surface. This makes it really tough to get rid of because it can survive regular flushing or cleaning processes. Once biofilm is established, it can lead to repeated contamination problems, which is why prevention is so important. It's much easier to stop biofilm from forming in the first place than to try to remove it after it's already there. This understanding is crucial for effective microbial control and maintaining clean environments. When it comes to preventing biofilm formation, there are several key strategies we need to focus on. First, using sanitary design principles is crucial. This means ensuring that equipment and surfaces are designed to avoid areas where biofilm could easily develop, such as sharp corners or crevices that are hard to clean. Continuous circulation of fluids also helps, as it prevents stagnation where bacteria might settle and form biofilms. Maintaining the correct velocity in pipes and minimizing dead legs, those stagnant sections where flow is minimal, is essential to keep the system clean. Additionally, controlling temperature and using ozone can effectively inhibit microbial growth. Applying validated sanitization procedures ensures that cleaning and disinfection are consistently effective. Finally, tracking microbiological data over time allows us to identify early warning signs of biofilm development, so we can take action before it becomes a bigger problem. Altogether, these measures form a comprehensive approach to preventing biofilms and maintaining hygiene. For pharmaceutical water piping, the most commonly used material is 316L stainless steel, especially for hot pharmaceutical water and water for injection systems. This type of stainless steel is preferred because of its durability, corrosion resistance, and ability to maintain purity under high temperatures. In some cases, high-purity polymers might be used instead, but this depends on the system's design and the method used for sanitization. When selecting materials, it's crucial to consider factors like extractables, how compatible material is with the water and other system components, as well as its ability to withstand the temperature and pressure conditions. Additionally, the material needs to be easy to clean to ensure the system remains free from contamination. All these factors play a vital role in maintaining the integrity and safety of pharmaceutical water systems. Question 26 asks why orbital welding is preferred for stainless steel loops. The main reason is that orbital welding offers repeatable and controlled weld quality, which is crucial for consistency and reliability. Additionally, it creates smooth internal surfaces, making the loops easier to clean and maintain. This is especially important in applications where hygiene is critical. Finally, orbital welding allows for detailed weld records, borescope inspections, and thorough documentation, all of which support good manufacturing practice, or GMP, traceability. These factors together ensure both quality and compliance in stainless steel loop fabrication. Let's talk about why surface finish is so important in a water loop. First, smooth surfaces play a critical role because they minimize places where organisms and residues can stick. This is crucial for maintaining cleanliness. A good surface finish also makes it easier to clean, sanitize, and drain the system effectively. On the other hand, rough or damaged surfaces can create hiding spots for contamination, increasing the risk of microbial growth and other issues. So, ensuring a smooth and well-maintained surface finish is key to keeping the water loop safe and hygienic. So, what exactly do we mean by a drainable system? Essentially, It's a system designed so that all the pipes, tanks, and low points can be completely emptied of liquid. This is achieved through careful planning of the slope, strategic valve placement, and an optimal piping layout. The goal is to prevent any water from being trapped inside, which is crucial because any retained water can create stagnant pockets. These stagnant areas can be problematic during maintenance or sanitization, as they can harbor contaminants or bacteria. So, having a drainable system ensures that the entire system can be thoroughly cleaned and maintained, promoting hygiene and operational efficiency. So, the purpose of a tank vent filter is to protect the tank from airborne contamination, especially during changes in the liquid level inside the tank. As the liquid level rises or falls, air moves in and out, and without a filter, dust, dirt, or other contaminants could enter the tank and compromise the contents. These filters are usually hydrophobic, meaning they repel water, which helps prevent moisture from entering. It's also important that the filter is properly rated for specific application, maintained regularly, and protected from getting wet. In some cases, the filter needs to be tested to ensure it's working correctly, keeping the tank's environment clean and safe. Now, let's talk about why sanitary diaphragm valves are preferred in certain applications. These valves are designed to minimize crevices in areas where liquid can get trapped, which is crucial because trapped liquid can become a breeding ground for microbes. By reducing these problem spots, diaphragm valves help maintain a higher level of cleanliness. They're also much easier to clean and sanitize compared to other valve types that don't have this design. This makes them especially valuable in industries where hygiene is critical, like food, pharmaceuticals, and biotechnology. Ultimately, these valves significantly reduce the risk of microbial contamination right at the points where the fluid is used, ensuring product safety and integrity. Now we'll be covering questions 31 to 40, focusing on microbiology, sampling, and monitoring. This section is crucial because understanding how microorganisms behave and how to accurately sample and monitor them forms the backbone of maintaining safe and hygienic environments. We'll dive into the methods used for sampling, the types of microbial contaminants commonly encountered, and how monitoring helps in preventing contamination and ensuring compliance with health standards. Make sure to pay attention to these concepts, as they are vital for effective microbiological control. When establishing a water sampling plan, it's important to focus on risk-based sampling points. This means selecting locations such as incoming water, pretreatment stages, reverse osmosis units, electroionization systems, tanks, supply and return lines, as well as critical points of use. You also want to include the farthest points from the source, areas with low water usage, and worst-case locations where contamination is more likely. The frequency of sampling should be tailored based on the qualification phase of the system, the level of product risk involved, and historical trends observed over time. This approach helps ensure a thorough and effective monitoring strategy. Now, let's talk about why it's important to monitor the return loop. The return loop represents water that has already traveled through the distribution system. By keeping an eye on this water, we can detect several critical factors. For example, we can identify any temperature loss, which might affect water quality. We can also monitor ozone decay, which is essential because ozone is often used for disinfection. Additionally, checking the return loop helps us spot microbial growth or any distribution contamination that could compromise safety. Overall, the return loop is a key point for controlled trending, giving us valuable insights to manage and maintain the system effectively. Here we have an important question about the difference between alert limits and action limits. An alert limit serves as an early warning that the system might be shifting or experiencing some variation. Think of it as a signal to pay closer attention. On the other hand, an action limit is more serious. It demands a thorough investigation, an assessment of potential impact, and the implementation of corrective measures if necessary. It's critical to not only define these limits clearly, but also to justify and track them over time to ensure the system remains in control and any trends are identified promptly. This approach helps maintain quality and prevents bigger issues down the line. If there's a microbial action level excursion, the first step is to notify quality assurance immediately. It's important to secure all relevant data and verify the conditions of both the sample and the lab to ensure accuracy. Next, assess a potential impact on the product by reviewing recent trends and looking at maintenance and sanitization records. Then, conduct a thorough investigation to identify the root cause of the excursion. After that, resample according to the standard operating procedures. Finally, implement corrective and preventive actions, or CAPA, to address the issue and prevent it from happening again. This systematic approach helps maintain product safety and quality. Why is sample flushing important before collection? Well, flushing helps remove any stagnant water that might be sitting in the sample valve or line. This is crucial because stagnant water could affect the accuracy of the sample, making it unrepresentative of the actual circulating water we want to analyze. By flushing properly, we ensure the sample truly reflects the current conditions. It's also important to follow the approved sampling procedure for flush time to maintain consistency and reliability in your results. Here we are looking at a common issue that can cause repeated high microbial results at a specific point of use or POU. Several factors might contribute to this problem. One possibility is a dead leg, which is essentially a section of piping where water stagnates because it's not circulating properly. Low local flow and infrequent use can also create conditions where microbes thrive. Additionally, a damaged valve can introduce contamination or disrupt normal flow patterns. Biofilm formation is another critical factor. Microbes can adhere to surfaces and create protective layers, making contamination persistent. Poor sanitation of the sample valve or contamination from hoses during sampling can also lead to misleading high microbial readings. To address these issues, it's important to review the system's design and how it's being used. Identifying the specific organisms involved and analyzing their trend patterns can guide corrective actions. Essentially, understanding both the physical setup and the sampling methods will help pinpoint the root cause and reduce those repeated high microbial results. The importance of online conductivity monitoring lies in its ability to provide continuous visibility of ionic purity in the system. This means we can constantly track the quality of the water or solution in real time. It helps detect crucial issues such as membrane failure, problems with the EDI system, chemical contamination, or valve leakage early on. By identifying these problems quickly, it supports rapid response, allowing us to address them before they impact other parts of the system more broadly. This proactive approach is key to maintaining system integrity and ensuring consistent performance. Online TOC monitoring is crucial because it allows for the rapid detection of organic contamination in the system. This means we can quickly identify if there are any issues such as cleaning agent carryover or organic fouling, which could affect the quality and safety of the process. Additionally, this type of monitoring helps detect abnormal conditions early, allowing for prompt corrective action. Importantly, online TOC monitoring doesn't replace offline laboratory testing, but rather complements it by providing continuous real-time data that can be used alongside trend reviews to maintain optimal system performance. Let's talk about how to trend microbiological data effectively. When analyzing this kind of data, it's important to look at several factors: individual data points, the time frame, the organisms involved, the seasonality, and the maintenance and sanitization cycles. By examining these elements, we can identify gradual increases in contamination, recurring isolates, or patterns that are specific to certain locations. Recognizing these trends early allows us to take preventive actions before any failures occur, ensuring better control over the microbial environment. This approach helps maintain safety and quality in the long run. Let's talk about objectionable microorganisms. These are organisms that could potentially pose a risk to a specific product or process. The level of risk they present isn't the same across the board. It really depends on several factors. First, the route of administration matters. For example, whether the product is ingested, injected, or applied topically can influence how dangerous a microorganism might be. Next, the patient population is crucial. Some groups, like immunocompromised patients, may be more vulnerable. Finally, the inherent characteristics of the product itself also play a role. To effectively manage these risks, we need to identify these microorganisms, monitor if they keep appearing, and assess the impact they could have. This helps us decide the right response to keep products safe. Now, We're moving on to the section covering qualification, validation, and good manufacturing practices, focusing on questions 41 through 50. This part is crucial because it deals with ensuring that all processes, equipment, and systems are properly qualified and validated, guaranteeing consistent product quality and compliance with regulatory standards. We'll also delve into GMP principles, which are the foundation for maintaining high manufacturing standards and safeguarding patient safety. Let's explore these concepts in detail to understand their significance and how they guide our manufacturing processes. So, let's talk about the difference between commissioning and qualification. Commissioning is all about making sure that the engineering installation is complete and that the system is operationally ready. It's like checking that all the pieces have been put together correctly and that everything is working as expected from a technical standpoint. On the other hand, qualification goes a step further. It provides documented evidence, following good manufacturing practice or GMP, to show that the system is suitable for its intended use in pharmaceutical production. Qualification involves a formal quality oversight process, ensuring the system meets all regulatory and quality requirements. Both commissioning and qualification need to be carefully planned and documented, but qualification carries an extra layer of formal validation to guarantee compliance and product safety. Let's talk about IQ, OQ, and PQ, which are key steps in the qualification process. IQ stands for Installation Qualification. This step ensures that the equipment has been installed correctly according to the approved design and all the necessary documents. Next is OQ, or Operational Qualification. Here, we verify that the equipment operates properly across all the defined operating ranges and that alarms function correctly. Finally, PQ, or Performance Qualification, demonstrates that the equipment consistently performs as expected under routine, real-world conditions. Together, these steps ensure that the system is properly installed, works reliably, and meets performance standards in everyday use. For a water system, there are several critical documents that we need to have in place to ensure proper design, installation, and operation. First, we have a user requirement specification and design qualification, which outline the system's intended function and verify that it meets the design criteria. Then there are the piping and instrumentation diagrams, specifications, and the factory and site acceptance tests, which confirm the system works as expected before and after installation. Risk assessments and detailed drawings help us identify potential issues and understand the system layout thoroughly. Material certificates and weld logs provide traceability and quality assurance for construction materials. Passivation records, calibration certificates, and standard operating procedures are essential for maintaining system integrity and operational consistency. Preventive maintenance plans ensure the system stays in good working order over time. Finally, installation, operational, and performance qualification protocols and reports, along with sampling plans, trend analyses, deviation logs, and change controls, document the system's validation and ongoing compliance. All of these together make up the comprehensive documentation required for a water system. Let's talk about what a user requirement specification, or URS, actually is. Essentially, the URS defines what the system must achieve. It's a detailed outline of the key criteria that the system needs to meet. This includes important aspects like the water grade, which sets the quality standards, as well as capacity, pressure, and flow rates. It also covers points of use, or POUs, along with monitoring and sanitization requirements. The URS is critical because it serves as a foundation for the system's design, qualification, and acceptance criteria. In other words, every stage from planning to final approval relies on the specifications laid out in the URS to ensure the system performs as needed. To validate a sanitization cycle, the first step is to define a clear method that includes all critical parameters. This involves specifying the exact conditions under which the sanitization should occur, such as temperature, chemical concentration, and exposure time. Setting acceptance criteria is essential to determine if the cycle is successful. Next, it's important to challenge the process by identifying worst-case points, the most difficult areas to sanitize, to test whether the cycle can effectively reach and sanitize those points. After that, verification of microbial recovery ensures that any potential contamination is eliminated. Finally, repeated documented runs are necessary to demonstrate consistent control of microbial contamination. This validation process assures that the sanitization cycle is reliable and effective under real-world conditions. Change control in a water system refers to the formal good manufacturing practice, or GMP, process used to evaluate and approve any changes. This ensures that any modifications don't negatively impact the system's performance or product quality. Examples of changes that require this process include adding a new point of use, replacing membranes, changing the programmable logic controller, or altering sanitization procedures. It's crucial to assess how these changes affect validation, documentation, staff training, and ultimately the quality of the product. This structured approach helps maintain consistency, compliance, and safety throughout the water system's life cycle. Calibration is crucial because it ensures that the critical instruments we rely on provide accurate and reliable data. Think about the key measurements we take, like conductivity, TOC, temperature, flow, pressure, ozone, and pH. If these instruments aren't properly calibrated, the data they produce could be misleading. This could lead to poor decision-making, incorrect assessment of deviations, and ultimately impact the quality and safety of product releases. So, maintaining proper calibration is essential to uphold the integrity of our processes and products. When preparing for a GMP audit, the first step is to verify that all current standard operating procedures are up to date and properly followed. It's important to check that all personnel have received the necessary training. Additionally, you should confirm that preventive maintenance, calibration, and validation activities are all current and well documented. Next, review recent trends, any deviations, and corrective and preventive actions taken, along with change controls, to ensure compliance and continuous improvement. Finally, ensure that all system drawings accurately reflect the installed equipment and that all records are easily retrievable. This thorough preparation helps demonstrate compliance and readiness during the audit. Let's talk about CAPA, which stands for Corrective and Preventive Action. Corrective action is all about identifying and eliminating the root cause of a problem that has already occurred. It's a way to fix issues so they don't happen again. On the other hand, preventive action focuses on reducing the chances of a problem recurring or even stopping potential problems before they happen. Together, CAPA helps organizations maintain quality and improve processes by addressing both current issues and future risks proactively. When performing a risk assessment for a water system, the first step is to identify potential failure modes and understand how they could affect both water quality and the quality of the product. It's important to look at how severe each failure could be, how likely it is to happen, and how easily we can detect it. To organize this process, we use tools like FMEA, which helps analyze possible failures, HACCP, which focuses on critical control points, or risk ranking methods to prioritize risks effectively. This systematic approach ensures we can design controls that minimize risks and maintain safety and quality throughout the water system. Now, let's focus on some troubleshooting scenarios. In this section, we'll go over questions 51 through 55, which are designed to test your ability to diagnose and resolve common issues. These questions will help reinforce your understanding of problem-solving techniques and how to apply them effectively. As we work through each scenario, pay close attention to the key details and think about the best steps to identify and fix the problem. This practice will be really helpful in developing your troubleshooting skills. Here we have a question about a sudden increase in conductivity after reverse osmosis. When this happens, there are several key areas I would investigate. First, I would check the conductivity levels of both the feed water and the permeate to understand the extent of the issue. Additionally, monitoring rejection rates, operating pressure, recovery, and flow rates can provide valuable insights into system performance. Next, I would conduct a thorough inspection of the membranes themselves, along with seals, O-rings, valves, and look out for any possible cross-connections that could be compromising the system. Finally, reviewing the pretreatment processes, chemical dosing, and any recent maintenance activities is crucial, since changes or lapses in these areas may contribute to the problem. By systematically checking these factors, we can identify the root cause of the conductivity spike and take appropriate corrective action. Here we have a question about a decrease in RO permeate flow. The likely causes include scaling, fouling, biofouling, or clogged cartridge filters. Other factors to consider are reduced feed pressure, low temperature, high differential pressure, or issues with the pump. It's important to review the trends carefully before deciding whether cleaning, sanitization, or membrane replacement is necessary. This helps ensure the right maintenance action is taken based on the actual problem. If the total organic carbon, or TOC, levels rise in the purified water loop, the first step is to check the status of the analyzer itself to make sure it's working correctly. It's also important to confirm these readings by conducting offline testing to rule out any false positives. Next, review recent cleaning and sanitization activities to see if anything might have contributed to the rise. Don't forget to check the status of the carbon bed and the upstream performance, since issues that can affect TOC levels downstream. Finally, assess if this increase has impacted the product quality. If the rise in TOC is confirmed and poses a risk, initiate a thorough investigation to identify and resolve the root cause. When responding to an endotoxin excursion in water for injection, the first step is to immediately notify quality assurance and take the affected water or its use under assessment. It's crucial to verify the test result to confirm the excursion. Next, review the microbial trends to identify any patterns or anomalies, along with assessing the sanitization procedures and the overall system conditions. Consider the potential impact on the products that may have been exposed. Then, work to identify and correct the root cause of the excursion. Before releasing any product, implement enhanced monitoring to ensure the issue is fully resolved and prevent recurrence. This systematic approach helps maintain product safety and compliance. When considering why you should hire me as a pharma water engineer, there are a few key points I'd like to highlight. First, I bring a strong combination of engineering troubleshooting skills and a thorough understanding of good manufacturing practices, or GMP. This means I don't just focus on the technical side. I also ensure everything complies with strict industry standards. I can support various critical functions, including operation, maintenance, qualification, validation, trend analysis, and audits. My top priority is always to maintain a reliable water system that safeguards product quality and, most importantly, patient safety. This holistic approach ensures the water systems not only function efficiently, but also meet regulatory requirements and protect the end user.